This conference will now be recorded.
A, a very good evening to one and all here. I am Priyanka, coordinator and faculty member of Biosetup Lifescience. On behalf of Biosetup Lifescience, take the honor to welcome our founder coordinator Arghyadeep sir, guest speaker Christopher sir, speaker Bhargavi ma'am, faculty member, and my dear participant to this webinar on the celebration of National Science Day. Biosetup Lifescience wishes everyone a
This
Bhargavi,
conference will now be recorded.
Faculty of Biosetup Lifesciences, and today we are conducting the Science Day. And on behalf of Science Day, we are conducting webinar on innovative research topics in life sciences by the Professor Christopher sir. Okay, now I am introducing the sir. It is a great honor to introduce and welcome our chief guest this evening. And on this day, here we are celebrating the Science Day. The main objective of National Science Day is to bring awareness to people about the importance of science in our daily life and to encourage people by popularizing science and technology. And every day, this day is celebrated with a particular theme. This year, the theme of National Science Day is Future of Science and Technology Innovation Impacts on Education, Skills and Work. As a student, it is our responsibility to honor great man and his invention by contributing greatly to the field of science through innovation. I wish all the science enthusiasts the very best to enhance their scientific zeal. Okay, now I am going to the topic about the topic and embarking on a PG course or accepting a postdoctoral position use it to be a step towards becoming an expert in one highly specialized area. For those who prefer to see the bigger picture and want the excitement of a more flexible and spontaneous career, looking beyond the confines of your old school disciplines may be just what the doctor ordered. And today's topic is innovative research topics in life science. As a life science, here many disciplines are there in life science like biotechnology, biochemistry, molecular biology, genetics, and plant tissue culture, genetics, all these are there. And nowadays, the research is mainly focused on interdisciplinary sciences. And this is where two or more disciplines combine to create more than sum of their part. It is the home of blue sky thinking. And some of the big challenges we face, both societal and scientific, are just not solved by people sitting in their single discipline field. Bringing those disciplines together in the long term is what provides the big breakthroughs. This is about the topic, today's topic, and that is, so we'll discuss about the interdisciplinary sciences. Okay, and next, regarding the source introduction, about Sir Dr. T. Christopher Rubin, sir, and Sir has done his PhD in botany, cytology, cytogenetics, and PhD from Kakatiya University in mutation breeding and tissue culture, and worked as research associate in University of Delhi, South Campus, New Delhi in plant biotechnology department, and he also worked as training associate in Krishi Vigyan Kendra, Karimnagar, Andhra Pradesh in agroforestry and also worked as Research Associate in Kakatiya University in Plant Biotechnology, and Sir also handled many positions in many universities. He worked as assistant professor in Kakatiya University, and also now he is working as a professor in Chaitanya Demutubi University, and also he is working as a dean research development in Chaitanya Demutubi University. And Sir also worked in many administration departments, and he has the teaching experience of 14 years and research experience of 19 years, and he has handled MPhil students of 1% and PhDs of 7%, and currently under his guidance the PhD ones are working of 3, and he also worked as a membership of academic bodies, and in that he worked in Indian Society for Genetics and Plant Breeding, New Delhi, and also worked in Society for Plant Biochemistry and Biotechnology, New Delhi. And next here, he also worked in as a member in Agricultural Horticultural Society, Hyderabad, Telangana Academy of Sciences, Hyderabad. Okay, and he is also a member of BOS in Biotechnology and Botany in Kakatiya University and Chaitanya Shatavahana University and Usmania University also. And he also attended international conferences of 6 and national conferences of 25. And he also visited Germany for exchange and collaboration between Indian and German scientists working in an area of plant evolution. And also he visited Professor Susan Renner's lab as a scientist and worked on chromosomes of Lupa and Briven. And he also published articles of 10 articles and organized workshops of 10, and done he has given the research abstracts of 20 and publications of 45 and major research projects he done of 2. This is all about the Christopher Rubin sir, and we are heartily welcoming the sir on behalf of Biosetup Life Science organization. Thank you, sir. Over to you, sir.
Thank you, madam. Thank you for all those good words that you have spoken about me. Especially, I thank Arghyadeep, the founder of Biosetup Lifesciences, and Bhargavi madam for giving me this opportunity. Right. The topic of our webinar today, national webinar, is innovative research in life sciences. So what are the topics that we have in life sciences? So I will be presenting it through this PPT. So I think all the participants can stop me in between or at the end, and then they can ask questions. Most of it is well known to you, but I am trying to present that what is the need of interdisciplinary work in life sciences. Okay, right. So yeah, today we are celebrating National Science Day on 28th February. This is Sir C.V. Raman's birthday. So on this day, we are celebrating it. He is the first Indian Nobel laureate for experimental studies. I repeat, he is the first Indian Nobel laureate for experimental studies, and he was awarded Nobel Prize for for Raman effect in physics in 1930. One of the tall personalities of science in our country is Sir C.V. Raman. So we respect his contribution, and we also appreciate his work towards the progress of science in our country. Right. The question is, what is research? Everybody think that what is that definition that we have for research? The word research is derived from a French word called researcher. That is to search with re-expressing intensive force. To search with re-expressing expressive or expressing intensive force. That is the term definition for research from its origin word. Right. What do we do in research? Research is we are trying to inquire, trying to examine facts and figures that add to the existing knowledge. In this endeavor, we integrate biological sciences, chemical sciences, mathematical sciences, engineering sciences, engineering and physical sciences with arts, humanities, and social science. So there is no scope left out wherein we don't interloculate with others. Every part of the study, every part of the system that we are living in is integrated into research. Okay, so it is a multidisciplinary resources to propose and develop innovative, enduring solutions and transform the latest scientific discoveries into new technologies. That is research. Already it is there, we are trying to search it out. Already it is there, we are trying to put forth our scientific methods, techniques, and equipment to find out what exactly is that. That is research. Okay, so in research we have two labs. One is called dry lab, the other is called wet lab. Everybody are very conscious that a mathematics department does not have a lab. These days it's not like that. The concept is a mathematics lab has geometry box. A mathematics lab has a abacus instrument, geometric figures, jigsaw puzzles, Rubik cube, and because of virtual lab these days we have a personal computer also. So this is called dry lab. Versus wet lab. Wet lab is your regular lab, that is where you have your equipment, instruments, chemicals, you have a microscope, you have an incubator, you have a physical balance. So whatever idea or concept that you have got in your dry lab will have to be demonstrated in the wet lab. So in the process of demonstrating, we do experimental work, we do field work. So that is what we call as wet lab. So any researcher who is interested to do experiments have to work in a wet lab. A mathematician may not need a wet lab, but they need these kinds of equipment. So there are two labs, dry lab and wet lab in research. Okay, right. There are three L's for a researcher. Any researcher, a postgraduate researcher who's performing a project, is interested to perform experiments for his project, or a PhD scholar is performing experiments. So if you are looking into this, we have three L's: landscape, laboratory, and library. These are mandatory for any researcher, right? What is landscape? Mother Nature is your landscape. Okay. In landscape, you have natural and artificial. Mother Nature is natural. Artificial is now these days they are developing islands. They are developing hillocks. That is artificial. Then in laboratory, again, Mother Nature is the laboratory. But we have developed laboratories. We call them as artificial laboratories. You have physical, chemical, biological, medical, herbaria, botanical gardens, arboretum, museum, fish aquariums, gene banks, agricultural feeds. All these are human-made. So you call them as artificial. So this is the laboratory. So you have a laboratory. Mother Nature is the first laboratory, and the artificial laboratory is the one that we have developed, man has developed. Then library. Mother Nature again is a library. But in the library that we have developed, we have books, journals, manuscripts, digitals, we have photographs, paintings, films, documentaries, etc. So you have three L's for this library that is artificial. So again, I repeat that there are three L's, that is landscape, laboratory, and library. In natural, it is the Mother Earth, Mother Nature that is natural, and we have developed over a period of time many artificial resources for a researcher. Okay, now an idea has to be formed. What is that we are supposed to do in research? So when you are supposed to do in research, which you can perform experiments with, we have 5 physical elements in our culture. We call it as Panchabhuta. Okay, so I have given them some titles here. Like the first physical element is the earth. As I told you, Mother Earth. Panchabhuta, you call it as Prithvi. Okay, then water, that is Jal. Then fire, that is Agni. Then air, that is Vayu. Then outer space with heavenly bodies is Akash, where you have sun, moon, planets, and stars, etc. So these are the 5 physical elements that where we can do our research. Like take for the example of Earth. What is that we are supposed to do here? The study of the Earth's development, how the soil is produced, called pedology. And the application of this particular science is called biology. What is that we need from the earth? We need food from the earth, right? We need fiber, we need shelter, we need medicine. So how do you do research? Improve the quality and the quantity of the food that is needed to human. So earth, water, fire, air, outer space is the boundary for us to perform our experiments, to do research. There is no limitation anywhere. Take for example the 4th one, that is air, Vayu. Okay, the study is animalogy, right? What is that application part we have? Atmosphere. What do we need from the atmosphere? Oxygen. We need carbon dioxide. Oxygen for respiration, carbon dioxide for photosynthesis. So what is that research that we are doing? We want pure air, pure oxygen. We want fresh air. We want fresh oxygen, right? So these are the areas where we can do research. We can do experiments, right? So 5 physical elements. Let us go to matter, right? These are the 5 physical elements. You see, this is the earth. This is the air that we are talking of, okay, and this is the Vayu. This is called Agni, right? This is the air. This is water. This is Jal, and this is the planetary bodies we call the outer space, right? All the 5 are depicted here, right? Now we have 4 fundamental states of matter: solids, liquids, gases. The new addition is plasma. Everybody are aware of this. Solids, liquids, and gases. Okay. So I've given you what is a solid, right? What is the definition of solid? What is definition of liquid? What is definition of gas and plasma? Plasma is made up of ions of gases, right? Atoms which have some of their orbital electrons removed and free electrons. What do? Earth's surrounding space environment, interior of sun, solar corona, stars, lightning, neon lights, all these are plasma, right? You have these days we have plasma TV also, right? So these are the 4 fundamental matters, states of matter, which we can perform research, right? Okay, we know that this water exists in 3 states: solids, liquids, and gases. Ice is solid, the vapors is the gas, ice is solid, water is the liquid, and then vapor is the gas. They are interrelated. One can change the other. Okay, right. Okay. Now coming to life science, there were only 2 classifications earlier. That is life sciences, where we study about living organisms, biological organisms. The other is physical sciences, where we study about non-living things, non-living aspects of it. But I've also included mathematical science. This is an abstract science. This is an abstract science. So I have 3 on this presentation: life sciences, physical sciences, and mathematical sciences. In life sciences, we have plants, botany, animals, zoology. Then in zoology, we have again humans, animals, and birds. Okay, what are the departments in zoology? Human, medical, and health science. Animals, cattle, and wildlife, that is veterinary science. And then birds is the poultry science. Then in case of plant science, we are interested in performing research into this because plants provide us with foods and nutrition. Microbes also are foods, right? So what is that area that we can develop technologies in? This is genetic engineering, genomics, proteomics. In case of medical and health science, we have therapies, in vitro fertilization, vaccines, drugs, and then how to diagnose a disease. This is how we discuss and study about life science research. Physical sciences are also very important for us, like physics and chemistry. In physics, we study the structural properties. In the chemistry, we study about chemical properties. The departments that have interdisciplinary are biophysics and geophysics. In chemistry, I've included forensic science. In technology, we have DNA fingerprinting. In biophysics and we have biosensors to detect COVID-19 virus. So COVID-19 is a non-living virus, but we need to apply physical sciences to find out the information about biosensors. Mathematical sciences, we have mathematics, where we have the algorithms, and the department is statistics. Then in technology, we develop models and modules. We develop models and modules for that. Computer science operates on binary system. What is important for computer science these days is robotics, artificial intelligence, bioinformatics. This is the technologies that are emerging from the area of computer sciences. Then in engineering, we have scientific principles and mathematics in this study. All branches of engineering, like we have designed and we have developed an aeroplane. We have constructed hanging bridge. We are sending into space shuttles, right? Electronic and electrical gadgets. We have developed metal alloys, and we have designed personal computers. Today, computer is every home-named term these days, right? Okay. Now, my question would be is, what is that I am trying to tell you is, there is no science which does not depend on other science. Biological science has to depend on mathematics. That is what I'm trying to tell you. I'll repeat. There is no science which is independent. Every science depends on the other. Then only we will be able to get some information for the society. What I'm presenting you is genetics. This is Mendel, he's father of genetics. In 1866, he developed this concept of the laws of genetics. Okay. On the right side, you have some numbers, some digits here. Because of his work for which he applied mathematics, he came up with these ratios. You call them as dihybrid ratio, okay, monohybrid ratio. Okay, this is a dihybrid ratio system. Okay, so no individual branch of science is separate. It has to depend, it has to draw information from the other branch of science. Okay, right. Now, using this information, this breeding, this information of genetics, Norman Borlaug produced Green Revolution, in which we got our food grains. So he was awarded Nobel Prize for this in 1970. 104 years later, Norman Borlaug was able to use the laws of genetics for improving crops and increasing crop productivity. Yeah, this is one of the greatest invention discovery that we can think of. That is, soldiers were dying because of infection. There were no chemicals to treat infection. In 1928, Alexander Fleming, he is a microbiologist. He is a doctor. Okay, he took fungi, and from them he isolated the first antibiotic called penicillin. And penicillin is now able to control infections and is used in all surgery cases. Yeah, this is another very important discovery that we can think of. 1953, the double helix model for DNA was proposed by Watson, Crick, and then in 1962, Crick, Watson, and Wilkins were awarded Nobel Prize. You have onto my right side, this is Madam Wilkins. She's Rosalind Franklin Rosalind. You know what is her discovery is, you see here a spiral kind of arrangement. She's a physicist. So she was responsible using X-ray crystallography to say that the DNA is helical. DNA is helical. Okay. Then length is 34 angstroms, diameter is 20 angstroms, spaces between two bases 10 angstroms. Mathematics, physics, chemistry. What did chemistry contribute? Nitrogen bases, adenine, guanine, cytosine, thymine, ribose sugar, phosphoric acid. What are they all trying to do? They're trying to help biology. What is that? They're trying to study DNA, which is the genetic material. So that means biology, chemistry, physics, mathematics are all involved in this process of the discovery of double helix model. So there is no independent branch of science which this does not develop. Okay, right. Yeah, 1978. This is another milestone in medical science. This is test tube baby. Okay, 1910. Reverend Edward was awarded Nobel Prize. Why? Because Patrick Steptoe died by that time. So the first test tube baby was Louis Joy Brown. This is the contribution that they have made. You know, today all test tube centers in our country are because of these two people. These two great people, Patrick Steptoe and Robert Edward. So this is another area where we can think of interdisciplinary research between medical science, okay, and then chemistry. They develop models, mathematical models they develop. It's not that easy for them to develop, right? Yeah, this is the first patent awarded to Professor Anand Chakravarti in 1980. He developed a genetically modified Pseudomonas putida. What is this Pseudomonas putida? Pseudomonas putida starts to eat oil from the oil spill that happened in the sea. Okay, 1980, right? This man is a microbiologist. He's working on oil chemistry, right? And then humulin. Humulin is the first rDNA product produced in 1982, and this is saving millions of people who are suffering from diabetes. Okay, in this humulin, pharmaceutical sciences are involved, biotechnology is involved, okay, chemistry is involved. Without these interactions between these scientists, it is not possible. It was not possible. Then gene therapy. I will not go into all these details. 1990 gene therapy. This is ADA, severe combined immunodeficiency syndrome. Yeah, 1996, the stem cell research where Ian Wilmut first cloned the lamb. Okay, we'll not go into all these details, right? Yeah, 2003 is a human genome project. Every gene of our chromosome is now sequenced, available in public domain. You can go to any bioinformatic site, website, GenBank. You will be able to see what are these sequences or a particular chromosome 1, chromosome 2, and all those things, right? In 2006, there is using this technology. They have developed in South Korea. They have developed, they have regenerated or incarnated a dog from the dead tissues of the dog. Okay, so that means you can clone dead animals also, and then get a live animal from that. Okay, this was done in South Korea, in Seoul, right? Okay, here I wish to show you one very important thing. Honeybee and the ridge gourd. Ridge gourd is your Birakaya, where you have male and female flowers. Okay, pollination is because of the honeybee. Without honeybee, no pollination. Without honeybee, no seed production. So we are performing some work on this. I will try to show you one very important observation. Here, the flower that you have now just seen is the male flower. Okay, this is the female flower. Now look at the behavior of the honeybee in this. Yeah, what we found in this study was, what did we find in this study was, the male flower will have to release pollen grain. Therefore, the honeybee is vibrating with its wings the pollen anthers, so that pollen grain is released. In the second case, because the female flower does not have pollen grain, it does not vibrate its wings. See, nature has given us so much information that how a honeybee collects pollen grain from a male flower and how it connects a nectar from the female flower. Okay, so this is science. This is observation science. Okay, right. So this is what we are trying to say, right? Now coming to interdisciplinary life science research. Okay, right. Now what is biodiversity? We study about plants and animal diversities. Biosensors, gadgets with biological recognition system. Bioinformatics, sequences of DNA, RNA, and protein. Bioremediation. Can we degrade plastics? Plastics are a menace now. Biochemistry is chemistry of biological organisms. Science and technology is biotechnology. Medical equipment and diagnostics is biomedical engineering. Okay, biofertilizers is organic farming. Biochips is miniature laboratories. Biologicals are pharmaceuticals and vaccines. Okay, right. Onto the left side, onto the right side, I've given you some. For example, like DNA fingerprinting. What are we doing with DNA fingerprinting? We need to develop barcoding. Genomics, okay, proteomics, metabolomics, in vitro mutagenesis. Not only the cloned DNA can be generated in a PCR, you can also mutate, you can also change a base in the cloned DNA. It is now possible to synthesize genes in the laboratory. We are able to produce recombinant proteins. It's called molecular farming. Cryopreservation is one of the greatest scientific area to be worked on. We need to conserve germplasm of microorganisms, plants, and animals. Telomere stability is important in cancer biology. So work is being done on telomerase activity. So like this, you go on enlisting. There are myriads of interdisciplinary works that we can perform in life sciences. Okay, I have recorded about 30 prospective areas. Okay, for example, medical and health sciences, objective is RNA virus. Who are the professionals involved? A doctor, mostly a virologist, a computer scientist, a chemist, a pharmacist, and a biotechnologist. What are they trying to do? They have to develop vaccine for COVID-19 mutant strain, not the normal one, mutant strain. Okay, already because we have COVID-19 vaccine available, but I am trying to tell you that so many people are needed to develop mutant strain COVID-19. Like this, you can go through all these lists that I have shown you here. Like you have parasites and vectors, vaccine for tropical diseases, especially malaria. Okay, malaria is a very dangerous disease now. Then cancer types, organ transplants, right? In organ transplants, doctor is needed, a virologist is needed, a pharmacist is needed, a biotechnologist is needed. What are they trying to do? From stem cells, can we regenerate kidneys? Okay, can we regenerate kidneys? Like antibiotic resistance, most of the bacteria today are resistant to antibiotic. No longer we use penicillin, but medicine, doctor, microbiology, pharmacy, biotechnologists, they are supposed to develop newer antibiotics that will kill the resistant bacteria. Like this, you can see a list of these cases, right? Just look at 10th part, 10th serial number. Postmortem. What is happening in postmortem? Doctor, a microbiologist, a biotechnologist, a pharmacist, and an engineer are needed here. Why? We need to use PCR. We need to use ELISA. We need scaling electron microscope to decide what is the cause of the death of a person. Nowadays, we are not using all these. We are using those crude methods that we have been working on for the last 100 years, 150 years, right? This is animal biotechnology, okay? Animal biotechnology, rDNA technology, again, biotechnologist, biochemist, chemist, a computer scientist are needed to develop recombinant protein. Then animal cloning, okay, animal cloning, veterinary science, we need avian bird flu vaccine. So again, microbiologist, biotechnologist, biochemist, and a veterinarian are needed to develop diagnostics for cattle, for birds, and all those things. Okay, right. Then we have biophysics, need to develop biosensors, neurologists, biophysicists, a computer scientist, and a mathematician. Artificial neural network and associative memory, an essential function of the brain. This is the area of research. Like this, I can mention you a huge list of all these important areas where we can perform interdisciplinary research, right? You have a microbiologist, okay, CRISPR technology can be applied for genomics, right? There are many unculturable microbes. You cannot grow them in a laboratory. You cannot grow them in a laboratory. So a microbiologist, a pharmacist, and chemist have to come together to grow unculturable microbes. And these microbes have novel bioactive compounds. Maybe our future antibiotics are available in these unculturable microbes also. Work has to be done on these particular aspects. Then I've given you plant biotechnology, okay, and then we have plant tissue culture, cryogenics, chloroplast DNA, okay. All the biologists, biochemists, physicists have to join together, and then we are supposed to develop what you call as research into these areas. So these are 30 that I've given you. I have tried to give you. So this will be a kind of a lead for us to take up research. Okay, right. I have put up some very wild questions. Okay, I think you are able to hear me. Yes, Priyanka, you are there.
Yes, sir. Yes, sir.
I have put up a very list of very wild questions, and this is research. Okay, first question is animal cloning a step forward to human cloning? Maybe everybody will not agree with me. Is animal cloning a step forward to human cloning? One. Is it possible to bring back to life the Egyptian mummies, woolly mammoth, or dinosaurs? How do dogs detect COVID-19 positive or negative in patients? Can we engineer genes of hummingbird into a goose? Will banana or potato light up the incandescent bulb, 0-watt bulb? Can we create a photosynthetic bacteria? Edible vaccines a reality for viral diseases? What happens if a cancer cell is provided with a wall made up of bacterial capsule? Will vampire bats spread disease in poultry and cattle? Do birds have biological clock? Can we substitute human leg muscles with wing muscles of Siberian cranes? How frog avoids skin infections? Are positively geotropic roots magnetized by Earth's gravity? Can we engineer fluorescent genes into avenue plants or trees? Can we apply egg membrane to burn patients? Genetically modified organisms can be used for producing electricity or not? Is global warming the cause of death of honeybees? What happens if a plant cell is fused to an animal cell? Can we engineer hibernation genes from polar bears into a gymnosperm? Does dog tick suck blood from humans? Is florigen a flowering hormone? Mobile vibrations endangered house sparrow in Telangana. Will seawater rise to engulf islands and coastal regions? Is afforestation and deforestation balanced? What is the mechanism of transport of water in plants against gravity? Does a bursting air bubble generate electrical energy? Is it possible to patch broken neurons? These are wild questions. You may not agree with me, but every researcher, whether he is presenting his project work, or he is working on project work, on a research work, or a PhD, every researcher should have wild questions. How and why? I have given you some lead, these kinds of wild questions. I can also show them. See, this is a place where they are trying to get electricity from banana, electricity from potato, and whether they will light up, you don't know. They are working on that particular aspect. You can also perform this experiment in your house. You have a banana plant, see whether we can do this. You need the help of a physicist, you need the help of an electrical engineer, you are a biologist. So 3 people have to come together, right? So this is one very important area that we should think and discuss, right? Genetically modified bacteria can produce electricity. Are we interested? Again, I've told you, biologists are there, microbiologists are there, electrical engineering engineer should be there, okay, a computer programmer should be there. All these things can be worked out. These are very wild, but they are being worked out. Okay, right. I've told you that your mobile vibration, my mobile vibration, has endangered house sparrow in Telangana. You don't find them. What happened? Because of the vibration, these birds are endangered. Maybe someday they become extinct, right? Anthropology. Here you have a mummified body of a queen of the pharaohs. Okay, here is a person who has brought out a 3D scan of this particular, and he has developed this particular face of this particular queen. Okay, and this is how they have developed a fair skin for this lady. Then, can amoeba become green? Someone said amoeba cannot become green. Amoeba can take up chloroplast. Amoeba can take up chloroplast and see whether chloroplast will photosynthesize. Okay, right. Today, you have new method of creating transgenic birds. I don't want to go into all these technologies and techniques that have been used, but today we are able to produce transgenic birds. Okay, now whether this is right or wrong is a different issue, right? Okay, every participant here, when you go home, please go through this particular movie. This is called Human Cloning. cloning, a very useful what you call as a movie where you can see how human organs are being cloned in live individuals. In live individuals, organs are cloned, and these organs that are cloned are put to insurance. Insurance is going to be a big business now with human organs. This is Island movie, the Island movie, Human Cloning. Please see this particular movie, right? Okay. So to the close, I'm quoting Dr. APJ Abdul Kalam, ISRO scientist and former president of India. The best brains of the nation may be found on the last benches of the class. Okay, so I acknowledge, thanks to Biosetup Lifesciences founder, Mr. Arghyadeep Das, and Ms. Bhargavi, the lecturer, and also my own teacher, my colleague in the Chaitanya Deemed University, Professor B. Srilata Madam, for giving me this opportunity to present this information to the participants today. And I can take any question. Anybody interested? I can take those questions. Priyanka, you're there?
Students, if you have any doubts, you can unmute yourself and ask sir.
Good evening, sir.
Good evening.
This is Srivalli speaking.
Yeah.
Sir, I'm enthusiastic to know the answer for the question which I have mentioned on the screen. That is, what if a cancer cell is replaced by the cell wall of bacteria?
Yeah. What happens if a cancer cell is provided
Yeah,
with a wall made up of bacterial capsule? Yeah. Yes. You want to know why you
yes,
need
sir.
to
I
have
want to know the answer for that.
See, cancer cells are the most
Yes, sir.
and fast dividing cells. Any normal cell
Yes, sir.
will divide and produce only 2 cells for within 24 hours. But a cancer cell, this rule does not apply. It goes on multiplying, maybe produce 8 cells, 10 cells, or whatever number. In this case, if you can encapsulate a cancer cell, it will not divide. Encapsulation.
Sir, there are also chances for. Sir, but there are also chances for the breakage, like we can degrade that in our body itself when we encapsulate it with the bacterial cell wall.
Yeah,
Like
which
we
can
have
happen.
enzymes for degrading the bacteria.
Yes, we can. See, you have in our intestines bacteria called E. coli.
Yes, sir.
Right? E. coli is not dissolved. E. coli capsule is not dissolved. E. coli capsule is intact. Though
Yes.
we have so many enzymes in our in the intestinal system, but still E. coli is there. Today I was discussing with my son about lactobacillus. You are consuming lactobacillus every day in the curd that we use, yogurt that we use. Okay? So the cell wall, or let us call it as a capsule, is a very hard structure. If you try to encapsulate cancer cells, I'm not an expert in this. Can we stop further cell division? Can we stop further cell division? Another very important thing is capsule will not allow food material to enter to the cancer cell. Called angiogenesis, you call that as angiogenesis. What is angiogenesis? There are blood vessels brought by the cancer cells, and then the cancer cells start to draw food material from those blood vessels. Now, if you encapsulate these cancer cells, they will not have connectivity with the blood veins or arteries, so they will not be able to draw food material. That could be one reason where we can hold it. So the cancer cell dies. Without food, cancer cell dies. as I've told you, this is a very wild question. Very wild
Yes,
question.
sir. Yes, sir. Thank you, sir. Thanks for the information.
You're welcome. You're welcome. Anybody else?
Dear participants, if you have any doubts, you can ask sir. Please unmute yourself. Or you can else provide in chat box. Okay, sir. Thank you
Yeah,
so much,
Priyanka, go ahead.
sir.
Yeah.
Yeah. Okay. And also, on behalf of Science Day, we conducted poster presentations. In that, we are announcing the results of that poster presentation. And the first prize goes to Srivalli, Kola Srivalli. And the second prize is going to Debanjana. And third prize is Baishali. Okay. And next, sir,
Yeah.
is that fine, sir?
Yeah, that's right. That's fine.
Okay. Okay. I would like to thank our guest speaker, Professor T. Christopher, sir, for enlightening us with their knowledge. And today's webinar was full of knowledge and interesting things. And it gave deep insights into the life science topics, different interdisciplinary topics, and also revealed some interesting topics like mutualism in between rich growth and honeybee, and also wild questions regarding the various topics in interdisciplinary sciences. And I'm sure the precious knowledge the Sir gave will definitely help us in our studies and in our research. And once again, I would like to thank for taking out the time from their busy schedule and enlightening us with their knowledge, sir. Thank you so much, sir. And I would also like to thank Organiser for giving permission to organize this webinar, and also I am thankful to Professor Srilata Madam for inviting the professor for conducting this webinar, and also I am thanking you to all the participants who attended and here for paying your attention and learning. Okay, thanking you, sir.
Thank you, madam. Thank you so much.
Okay,
Thank you so much. See you again. Bye.
okay, thank you, sir.